Hello, everyone, and welcome to the ARO seminar series session titled Observations During the Study of Hair Cell Mechanotransduction. My name is Artur Indzhykulian. I'm an assistant professor at the Department of Otolaryngology of Harvard Medical School. My lab is located at the Massachusetts Eye and Ear Infirmary, and we study the sensory cells of the inner ear, the hair cells, aiming to learn more about their function and dysfunction, and also develop gene therapy tools to treat deafness and blindness. I'm pleased to be moderating today's webinar. Our presenter is very excited to host this webinar for you today. Here's a little bit about Dr. Peng. Dr. Anthony Peng is an assistant professor in the Department of Physiology and Biophysics at the University of Colorado. His lab is focused on studying the mechanisms of how the auditory sensory hair cells, the fastest mechanoreceptors in the body, transduce sound into electrical signals for the brain to process and interpret. To study the fast mechanical responses of hair cells, Dr. Peng developed high-speed stimulators and high-speed imaging techniques to capture the kinetics of these exquisitely fast mechanoreceptors. Prior to starting his lab, Dr. Peng trained at Stanford University and in the Speech and Hearing Bioscience and Technology program at Harvard and MIT. Dr. Peng lives in Denver, Colorado with his wife and 2 kids. Outside of the lab, he enjoys the outdoors of Colorado, including paddleboarding, hiking, climbing, and snowboarding. Before getting started today, we would like to review a few webinar tools that you as an attendee will be using throughout the presentation. These tools are located on the bottom of your screen. To keep questions from becoming distracting to our presenters, please utilize the Q&A function. You are welcome to submit your questions at any time throughout the presentation by typing into the Q&A function and clicking the send button. Please consider including your name with your question so that we may contact you with an answer should we not get to your question during the live session. Any questions that are not addressed during this session will be answered by the presenter in a follow-up email. Please note that your name and questions will not be visible to other attendees. Closed captioning for this webinar is also available for your use. This session is being recorded and will be made available for additional viewing. We hope you will also participate in our post-webinar evaluation so we can gather feedback, as this is our first webinar series. Additionally, the next ARO seminar series discussion will be presented by Dr. Jennifer Stone and moderated by Dr. Mark Warkol in November. More details regarding that talk will be available shortly. Lastly, if you are having technical difficulty, please email headquarters@aro.org for assistance. And now, without further delay, I'd like to introduce to you Dr. Peng.
Thank you, Artur. Thanks for the introduction, and thank you to the ARO Seminar Committee for giving me the opportunity to actually talk to all of you today. Today I'm going to talk about some of my work studying hair cell mechanotransduction, and actually one more thank you to Dr. Brownell, who actually gave a very nice introduction to the peripheral auditory system, which is what I'm going to go into further today as well. So what is, all right, there we go. Why is it that we study, or why is it that I study the auditory system? And the reason that I like to study the auditory system is because it's quite remarkable. It encodes a large dynamic range of sound intensities that span things that are as quiet as a pin drop to things that are as loud as a fighter jet launch. And this spans over 12 orders of magnitude. Additionally, our ear has an amazing frequency selectivity. So 2 keys on the piano differ by 6% in frequency, and our ears are able to easily discriminate that because we can discriminate down to just 0.2% differences in frequency. And so these 2 are features that the peripheral auditory system actually provides to the central auditory system that then allows us to process complex sounds such as speech, which is composed of many different frequencies that vary over time. And additionally, it allows us to pick out a given sound source in the presence of many other sound sources, known as the cocktail party effect. And so today, I'm going to go into how is it that the dynamic range and frequency selectivity, and looking at processes that contribute to the dynamic range and frequency selectivity of the auditory system. And so I'll start out with an introduction to the hearing and mechanotransduction mechanisms. And I'm actually going to talk about two major observations that I've made during my career. The first is regarding fast adaptation, and the second is regarding slow adaptation. And finally end with the summary and speculation of what these mechanotransduction mechanisms look like now. So let's just start into it. So sound enters the external ear and impinges upon the tympanic membrane. So the sound pressure waves are then converted into a mechanical vibration that then is passed through the middle ear bones and ends up in the stapes. The stapes then creates a fluid vibration within the inner ear structure. So this is the inner ear structure. Today we're going to focus predominantly on the cochlea, but the vestibular system is also a part of the inner ear, and there will be some studies that I talk about that involve the vestibular system. So if you take a slice through the cochlea, this cochlear structure, then you can see that the cochlea is composed of multiple fluid-filled compartments that spiral up along the cochlea. And in the middle of that structure, or in the middle of these fluid-filled compartments, sits the organ of Corti. So the organ of Corti is in this region and spirals up along. And so if you focus in on the organ of Corti, which is the sensory organ of the auditory system, then you have these sensory hair cells. In this case, there's two types of hair cells. There's inner hair cells as well as outer hair cells. And these cells are named because of the apical hair-like protrusions that sit on the apical surface. And so I'll refer to these hair-like structures as the hair bundle. And so the hair bundle is what actually converts the mechanical vibrations. So the mechanical vibrations in the fluid create a vibration within the basilar membrane. And this vibration within the basilar membrane then leads to a shearing of the actual hair bundles. And so the hair bundles then detect this shear and actually convert that mechanical energy into an electrical signal that can then be passed along to the central auditory system. And so if we focus in on the hair bundle, which is what I study, the hair bundle is responsible for the mechanotransduction process. And so when we say mechanotransduction, we solely mean that this is transducing, meaning converting the mechanical energy into an electrical energy. And so these hair bundles are very beautiful, and they have a very stereotyped structure where you have multiple rows of stereocilia. And these rows of stereocilia are graded in height to create this staircase architecture. If we take a slice through the side of one of these stereocilia, then you can get the idea, or you can see how the mechanotransduction process is thought to work. So these adjacent rows of stereocilia are connected by what we refer to as a tip link. So this tip link has some amount of tension in it, and that tension is transmitted to the channel, such that when we deflect the stereocilia, the tension increases in the tip link and then is detected by the channel to allow the channel to open and ions to flow into the hair cell. And this flow of ions then obviously signals the hair cell that some sort of mechanical displacement has occurred. And so the way that we study this process in the lab is using patch clamp electrophysiology. So this is a low magnification image, and we can see we have the patch pipette coming in from the side as well as the stimulator coming in from the bottom here. And so the patch pipette is basically a glass pipette, and what we do is we seal onto the basolateral side of this hair cell, which is the lower part of the hair cell. And then once we create a seal with the membrane on the hair cell, we actually rupture the membrane piece that sits within the within the micropipette. And this then allows us to create an electrical connection between the inside of the pipette and the inside of the hair cell. Thus, any electrical current that flows into the cell, then we can now detect in our patch electrode. Okay? And so if you look at a higher magnification image in this region, then you can see that there are multiple rows of these hair bundles. And so these are the outer hair bundles. The inner hair bundles are located over here. And in this case, we're using a stiff glass stimulator. And this glass stimulator is designed to fit within the hair bundle to deliver the displacement. So we bring in a displacement stimulator, or a mechanical stimulator, to actually cause a mechanical displacement such that we can then record the currents resulting from that mechanical displacement. Additionally, we can also have a apical perfusion pipette, and this allows us to control the extracellular solution surrounding the hair bundles. So when we do these experiments, we have a few characteristics that we see. So first is that when we stimulate the hair bundle towards the tallest row of stereocilia, so this is a step-like displacement, What we record in our patch electrode is a rapidly activating inward current. And then you see this inward current then decays over time. And this decay is due to a process called adaptation. Now I want to note that the convention in the field, and the convention in electrophysiology in general, is that whenever you have positive ions flowing into the cell, this is seen as a negative current. If positive ions are flowing out of the cell, then it'll be seen then as a positive current. Okay, so in this case, since positive ions are flowing into the cell and depolarizing the cell, we have a negative inward current. So we can do this for many different step sizes to generate what we call a family of curves. And then we can take the maximum current achieved for each displacement size and plot that. So this is now we're plotting the displacement on the x-axis and the relative or the normalized maximum current. And this generates what we refer to as an activation curve. Now the activation curve then describes the sensitivity of the channel before any actual stimulation has occurred. And so I mentioned the process of adaptation. And adaptation is defined as a decrease in current during a sustained stimulus. You can see the stimulus is not changing, but the amount of inward current has now decreased. Now the key feature of adaptation is that if we stimulate the hair bundle further, we can actually recover any current that had been lost. So we can recover that adapted current. So we can take this a step further and actually generate a full activation curve both before and after, in a sense, pre-displacing the hair bundle. So what we do is we move the bundle back and forth to generate an activation curve. We put on this displacement step and pre-displace the hair bundle, and then now move the hair bundle again to look at what's happened to the activation curve. And what you see then is you can see that with that pre-displacement, it actually shifts the activation curve to the right. And so this has important implications in terms of what this adaptation process can do. So the first is that it can contribute to the dynamic range. Because you can imagine in the first condition that the range of stimuli that is able to be detected by the hair cell is very narrow, only this much. But then as you adapt to various stimuli, then you can adapt and start to detect another range of stimuli. And so this then can allow us to increase the dynamic range. Secondly, it controls the operating point, and this basically tells us what range of displacements is the hair bundle actually sensitive to. So is it sensitive to mainly louder sounds, for instance, or sensitive to mainly quieter sounds? And the last thing that it can do is actually participate in frequency tuning. And so I'll talk about this a little bit more later. But there's two manifestations of adaptation that I want to point out. The first that we're going to often refer to is current adaptation or current decay. And so this is the decline in current during the sustained stimulus. And the second manifestation is that there's this shift in the activation curve as seen here, as demonstrated here. And this will be referred to sometimes as an adaptation shift or an activation curve shift. So when I started studying the mechanotransduction process, there had been a lot of work done previously. And the prevailing model of how this worked in the field looked like this. So again, we had the tip link that connected the adjacent rows of stereocilia. And then we had mechanotransduction channels located on either end, on both ends of the tip link, that was able to detect the tension within the tip link. And then finally, there were myosin motors that were located along the sides of the stereocilium. And these myosin motors were thought to then climb and slip and move basically along the sides of the stereocilium to adjust the tension in the tip link. Because you can imagine that is if that tip link was actually inserted or connected at a lower point, then there would be less tension in this tip link. Whereas if this tip link was connected higher and pulled up by these myosin motors, then the tension within the tip link would be actually higher. So there are two modes of adaptation that had been described as well. The first was the slow or motor adaptation process. And this is where calcium came in and actually bound to the myosin motors to cause the slipping of the myosin motors down the sides of the stereocilia in order to release tension in the tip link and allow the channels to close. The second mode of adaptation that had been described was fast or channel adaptation. Now this happened on a much faster time scale, hence the name. So slow adaptation happened on the order of 10 to 100 milliseconds, whereas fast adaptation occurred on the order of 0.3 to 5 milliseconds. And the idea here was that calcium was also involved and important and required to come through the channel and bind to some site near the channel. So just before I kind of started in studying mechanotransduction specifically, it had been discovered that at least in the mammal, the mechanotransduction channels weren't actually located along the sides of the stereocilia. This didn't change much for fast adaptation because fast adaptation could still occur where it went through the channel and bound to some site near the channel. Slow adaptation, on the other hand, no longer had a direct source of calcium near the myosin motors, and calcium would have to come from a more distant site, perhaps even mechanotransduction channels that are located at the tips of this stereocilia. Okay, so I'm going to go into the next portion of my talk, which is the first observation regarding fast adaptation. So when I first started studying mechanotransduction again, I was actually very interested in the frequency tuning mechanism. And the mechanotransduction process can actually act as a frequency tuner. And the way that this is thought to happen is that the adaptation process acts as a high-pass filter. And the reason this is because if adaptation can decrease the response to a given stimulation, then very slowly changing stimuli may be adapted away before it actually results in a response. And therefore, you could get a very small response for low-frequency stimuli. On the other hand, if the stimulation is very high in frequency, then these things are changing rapidly, and adaptation doesn't have a chance to actually decrease the response before the stimulus has already changed. And therefore, high frequencies have a large response. And so this then acts as a high-pass filter. On the other hand, the activation kinetics of the channel act as a low-pass filter. And that's because the channel has to actually open in order to allow ions to flow in. So very slowly changing stimuli would give the channel enough time to open and detect the signal. Whereas if the frequency is very high and the signal is changing rapidly, then maybe the channel cannot actually activate before the stimulus has changed. And therefore, activation kinetics act as a low-pass filter. And so if you take these two filtering characteristics and you combine them, then you get what's called a bandpass filter. The bandpass filter is important because then it selects out a narrower range of frequencies that the hair cell is actually able to detect. And this was seen to be the case in the turtle hair cells. So it was found that in low frequency hair cells, they had activation and adaptation kinetics that actually centered this bandpass filter at lower frequency, whereas higher frequency hair cells located in another portion of the auditory papilla was tuned towards detecting higher frequency stimulations. And so when I first started out, I wanted to see whether or not this was the case in the mammal. We knew that the adaptation kinetics had varied along the cochlea, suggesting that this could be contributing towards the frequency tuning of various hair cells. But what we didn't know was the activation kinetics, because the activation kinetics in the mammalian system were really fast. And so we had developed stimulators to try to look at whether or not we could faithfully see the activation. And so so this is an example trace of a recording that I had taken early on. And what I had noticed was that you started to see adaptation happening very quickly. And what I was afraid of is I was afraid that this adaptation was actually bleeding into the activation, such that it was potentially happening before the channel had a chance to fully activate. So what I wanted to do was actually get rid of adaptation. So then to do that, I turned to what we knew about adaptation. So what we knew was that adaptation depended on calcium. And so we can take advantage of the membrane potential and depolarize the cell in order to manipulate how adaptation would actually function. So at negative holding potentials, when the cell is at a negative potential, then this negative potential will attract, for instance, the positive ions. And so calcium would have a strong electrochemical gradient to be driven into the cell. And so since calcium is entering the cell, then we would be able to observe adaptation. So in this case, we would be able to observe adaptation, for instance, in the current response. Now, if we depolarize the cell and make the inside of the cell very positive in order to repel the calcium ions and actually start to drive them out of the cell, so either drive it out or at least decrease the rate of calcium influx into the cell, then we should be able to decrease the amount of adaptation. And indeed, that's what's predicted, that we would decrease the amount of adaptation if we go to a positive holding potential. Another thing I want to note is because, again, we talk about positive current is current that flows out of the cell. And so in this case, we have positive ions that are being driven out of the cell at positive holding potentials. And therefore, the direction of these currents changes such that we get positive currents now, okay? And so we did this then in the mammal to try to get rid of adaptation so that we could see the activation kinetics. And so we did this by depolarizing the cell. And what we found was that adaptation seemed to be robust both at negative and positive potentials. Now this is very different from what was observed previously. So these are, one other thing I want to note is these are 5 millisecond steps. So this step size makes it such that we're looking predominantly at the fast adaptation process. Okay, so 5 millisecond step. And so with a comparable 5 millisecond step in the bullfrog, people have shown that the Corey group had shown that there's adaptation at negative potentials, and that was actually lost at positive potentials. Okay, so this was peculiar, and this was an interesting observation because it was counter to what we had expected. So we wanted to investigate this a little further, and so we wanted to alter adaptation in a different way. So what we turned to then was changing the extracellular calcium concentration. So remember again that adaptation occurs when calcium enters the cell and binds to some site within the cell. Therefore, if we lower the extracellular calcium concentration, then we will lower the rate in which calcium actually enters the cell because we decrease the chemical gradient of calcium. And so what was found previously was that in high extracellular calcium concentrations, you could see robust adaptation. And as you lowered the extracellular calcium concentration, then the amount of adaptation decreased or even possibly went away. So we repeated this same type of experiment in mammals and found that indeed at negative potentials, we could see at negative potentials with high amounts of calcium, we saw robust adaptation. But as we lowered the extracellular calcium, we still saw the adaptation persisted. Again, this is suggested then that maybe calcium was not actually driving this adaptation process. So to do one other confirmation, we went back to depolarization. So I mentioned that the second manifestation of adaptation was the shift in the activation curve, the adaptation shift. So at negative potentials, when calcium would be coming in, we would expect that the activation curve would shift. But at positive potentials, when calcium's not coming in, then we would expect that there would be at least a decrease, if not no shift in the activation curve. So we did this experiment in the mammal, and again, at negative potentials, we found that the activation curve shifted. And at positive potentials, the activation curve still shifted, and to a comparable extent, suggesting then again that the fast adaptation may not actually be driven by calcium. And so these experiments, along with some others that I'm not actually going to talk about today, led us to the conclusion that the fast or channel adaptation process is not actually driven by calcium, as previously thought. Okay. So now I'm going to go into the second part of the talk, which is some of our observations regarding slow adaptation. So for reasons I'm not going to go into today, we started exploring a second mode of stimulation. Everything that I've talked to you about today up until now has involved what we've referred to as a stiff probe stimulator. Stiff probe stimulator is a glass pipette that's connected to a piezoelectric stack. And we can deliver a voltage to the piezoelectric stack, and this causes a given displacement of the glass probe. Now the glass probe is attached to the hair bundle, therefore this is delivering a displacement to the hair bundle. A second commonly used stimulation in mechanotransduction is referred to as the fluid jet. Now, the fluid jet similarly uses a piezoelectric device, but in this case the piezoelectric device changes the pressure on the backside of this fluid-filled pipette, which then causes a jet of fluid to essentially shoot out of the pipette or be sucked into the pipette. And so in this case, we're using that jet of fluid, this stream of liquid, to stimulate the hair bundle. And so these are inherently different types of stimuli. In this case, we're delivering a displacement to the hair bundle. And in the fluid jet case, we're actually delivering a force to the hair bundle since we're using the fluid as our stimulator. And so if you go back to high school physics, you'll know that force is related to displacement by this term K. So F is equal to Kx, and this K term is referred to as the spring constant and describes the stiffness of an object. So you can imagine if you're using your garden hose and you shoot the garden hose against the tree, you can see the tree doesn't move very much because the tree is very stiff. Whereas if you shoot a piece of paper or a blade of grass, the stiffness of that is much lower, therefore you're going to get a much larger motion. Okay, so the hair bundle is even more complex than what I've just described because the hair bundle is actually known to change its stiffness during stimulation. And that was shown by Ian Russell and colleagues back in 1992 in the mammalian hair cell, where they used a flexible fiber to stimulate the mammalian hair bundle, and they saw that the hair bundle continued to move during the stimulation. Now, this continued motion is actually referred to as a creep, as a mechanical creep. And this was confirmed later also using a fluid jet stimulation by Croce and colleagues, as well as by Berg and Fetty-Place using a flexible fiber in other mammalian hair bundles. And so because the hair bundle changes its stiffness, it's important to actually measure the motion of the hair bundle when we're using a force stimulation. And so originally, the technique that was using a dual photodiode system. And a photodiode is basically a light detector. And so it could detect the movement of a strip of light. In this case, the hair bundle acts as a strip of light as it moves across these two photodiodes. And so this type of experiment is very difficult because of two reasons. Number one, you have to align this strip of light, or meaning the hair bundle, precisely in between these two photodiodes to get a very good signal. And secondly is every time you do the experiment, you actually have to create a calibration where you actually move the hair bundle and a known distance to be able to know what the electrical signal is compared to the actual motion. And so this calibration and precise alignment makes this experiment extremely difficult. And these are two orientations that photodiodes have been used on the mammalian hair bundle. And so we wanted to come up with a different way because of the cumbersomeness and also difficulty of this experiment. So we turned to high-speed video imaging. And so we took advantage of advancing camera technology, which allowed us then to take videos of the hair bundle at 10,000 frames per second, all the way up to at least 50,000 frames per second. And so this made the experiment much easier and had some other advantages. So number one, we no longer had to have precise alignment because we could see the hair bundle, and we just had to be within the field of view of the camera. The second thing is that we no longer needed a calibration every time because we know the number of nanometers per pixel, and therefore we can then get a displacement very easily. And the third benefit of this is that now we can see the entire hair bundle. We no longer have to focus on the motion in a given direction. We can actually measure the motion in 2 dimensions here. So we started these experiments, and then we first had confirmed that when we put a flexible fiber in view, and we know a flexible fiber does not change its stiffness, we can confirm that the hair bundle does indeed change its stiffness during the stimulus. So you can see here that the flexible fiber motion in the same field of view has a square step, just as we're expecting with our force stimulus. However, the hair bundle exhibited this mechanical creep. Now, another interesting observation we made was that this mechanical creep seemed to mask the fast adaptation process. Now, what I'm showing you now is a 50 millisecond step. So this is 10 times longer than the steps that I showed you previously. And so with this then, the current declines that we're observing here are dominated then by the slow adaptation process because of the longer time scale. And so this allowed us to essentially focus on the slow adaptation mechanism and allow us to more easily see or visualize what was happening to slow adaptation. So during these experiments, we had seen that indeed at negative holding potentials, we could get very robust slow adaptation. And then when we depolarized the cell and changed the flow of calcium, then we lost that slow adaptation. This then fit well with the idea that slow adaptation was calcium dependent. The interesting observation, though, that we made in this scenario, in this case, was that the displacement did not change. And the reason that this is important is because that it was previously thought that the mechanical creep that's observed here is associated with the slow adaptation process. And so I'm going to explain that now. So the experiments done by Howard and Husbeth, and these are landmark studies using a flexible fiber on the bullfrog saccular hair cell. And what they did is they delivered a mechanical force step using the flexible fiber, and they observed exactly what we see, which is this mechanical creep in the hair bundle. And then they were simultaneously recording the mechanotransduction current, and they could see then that there was indeed both fast and slow adaptation in this case. Okay. And what they observed was that this hair bundle creep had a similar time course to the slow adaptation. And therefore, they posited that this mechanical creep was causally related with the slow adaptation process. So from this observation, the motor model of adaptation was developed. And so I'm going to explain the motor model of adaptation in more detail here. So again, the observation is that this mechanical creep matched with the slow adaptation process. And therefore, these two could be causally related. Now, an important piece of information to try to help understand why this is the case, or why the mechanical observations fit with what we're talking about, is to know that in a normal hair bundle, if there's no tip links, let's say we have a given stiffness of the hair bundle. But as we put the tip link in, if we put the tip link into the hair bundle, the tip link starts to tension, and the tension in the tip link actually contributes to the overall stiffness of the hair bundle. And we know that if we remove the tip links from the hair bundle, then we lose about half of the stiffness of the hair bundle. So this indicates to us that the tip link contributes roughly about half the stiffness of the total hair bundle. Okay, so then the motor model then suggested that at rest, we have our regular configuration of tip links in the hair bundle. And then once we stimulate, we increase the tension in the tip link. And then the idea is that when calcium flows in through the channel, this was going to cause these myosin motors to slide down along the sides of the stereocilium. And by sliding down the side of the stereocilium, then you release tension in the tip link. This reduction in tension in the tip link then will reduce the contribution that it has to the total hair bundle stiffness. And therefore, the hair bundle will continue to move in the positive direction because we're decreasing the stiffness of the hair bundle. And so it was thought then that this decrease in tension within the tip link could explain both the hair bundle creep, and also, since the mechanotransduction channels detect the tension in the tip link, that if the tension in the tip link is decreased, we would also get the closing of the channels and a decrease in the mechanotransduction current. So this model was very nice because it could explain both of these phenomenon. And so other studies also told us that, as I mentioned earlier, that calcium mediates slow adaptation. And additionally, that myosin motors were required for this process. And particularly myosin 1C was posited to be this adaptation motor that was climbing along the sides of the stereocilium. And so with this model then, the prediction would be that if we have large amounts of adaptation, then we would expect to see a good amount or a large amount of creep. If we were to decrease then that amount of adaptation, we should see a concomitant decrease in the amount of creep. And so that's exactly opposite though, or that does not fit with what we actually had observed, or the observation that I'd shown you earlier, because we can decrease the amount of adaptation by depolarizing, but we see no change in the actual mechanical creep. So we went to another method to confirm this, and we used sulfate and vanadate. So previously, Yamoa and Peter Bar Gillespie had used intracellular sulfate and intracellular vanadate to inhibit myosin motors, and showed that there was a decrease in the amount of adaptation. And indeed, that's what we find, that as we put these pharmacological agents intracellularly, we see a decrease in the amount of adaptation. However, when you look at these mechanical traces, you can see that the amount of creep doesn't look to change. Another way that we can actually manipulate the amount of adaptation is changing the intracellular calcium buffer. So intracellular calcium, calcium buffers in general bind to free calcium. And so you can imagine that if there's a bunch of intracellular buffer, then as calcium enters the cell, then the buffer would bind up that calcium before the calcium is able to reach its effector site, and in this case, mediate adaptation. And so as we increase the amount of intracellular calcium buffer, then we should decrease the amount of adaptation as well as slow the rate of the adaptation. And that's indeed what we find, that if we have a low amount of calcium buffer, in this case, we're using BAPTA, so BAPTA is a calcium buffer. And so low amounts of the calcium buffer resulted in good amounts of adaptation. And as we increase the amount of calcium buffer, we saw a decrease in the amount of adaptation. And additionally, we also see that the slowing of the adaptation process as seen here. And again, summarizing that increasing the amount of intracellular calcium buffer decreases the amount of adaptation. And also at positive potentials where we don't expect any adaptation, we see similarly low levels of adaptation. Okay, so now we have multiple ways to actually manipulate the amount of adaptation. And so we wanted to then look at the hair bundle creep and quantify it. And so the way that we quantify the hair bundle creep is we fit the creep with a double exponential decay. And so you don't have to know about the details of this equation. I'm going to bring out 3 parameters that we actually quantify and focus on today. The first is the time constant of the slower creep. And the reason that we chose the slower time constant is because this is what matched the slow adaptation time constant most closely. The second parameter that we're going to look at is the percentage of the slow creep. Now this gives us an idea of the relative magnitude of the slow creep as compared to the total creep. And the final thing that we're going to look at is another measure to give us an idea of the magnitude of the creep. And so this is the magnitude of the total creep relative to the total step size that the hair bundle exhibits. Okay, so we can now do this, we can now quantify this motion. And so with low amounts of BAPTA, this is where we have the highest amounts of adaptation. We see that the slow time constant of the bundle matches pretty closely with the slow adaptation time constant. And then now as we depolarize the hair bundle or increase the intracellular BAPTA, these are all things that decrease the amount of adaptation with sulfate as well as vanadate. In all of these scenarios, there's very little change in the motion of the hair bundle, suggesting then that, and this is then further quantified by the parameters that I mentioned earlier, and that there's no change in these parameters, suggesting then that the hair bundle creep is not actually correlated or causally related with the slow adaptation process. So you could argue that, well, we're doing these experiments differently than what was done previously. So we were using a fluid jet, whereas previously they used a flexible fiber. Additionally, we're using this on cochlear hair bundles, which have a very different structure than the vestibular type hair bundles that were used previously. So this is a mammalian vestibular epithelium, the utricle. And so the hair bundle, you could argue that the mechanics due to this cohesive hair bundle could be different than the mechanics with this U-shaped bundle. So we wanted to repeat these experiments then, using a flexible fiber on the mammalian vestibular system, which has this cohesive hair bundle. So this was the previous experiment that was done in the bullfrog, so we wanted to mimic that. And so in this case, we're using a gerbil utricular hair cell. And this line just indicates where we're going to measure the motion of the hair bundle. And then there's actually a flexible fiber in here that's extremely difficult to see because of the thinness of the fiber. So we stimulate the hair bundle with the flexible fiber, and we indeed see that there's both there's slow adaptation at negative potentials, but that slow adaptation is lost at positive potentials. And then we can simultaneously then measure the motion, and we can see that the motion doesn't change very much. And we can highlight then, I'm going to highlight a stimulus step size of roughly the same magnitude. And you can see that in this case, there's a lot of adaptation at positive potentials. And I'm just flipping the current here so that it's more easy to visualize the difference. So we have lots of adaptation at negative potentials, but not much at all at positive potentials. And the hair bundle motion, however, remained largely the same. suggesting again that the hair bundle creep is not actually correlated with the slow adaptation process. The final experiment I'm going to talk about today is with myosin 1C. So myosin 1C, I mentioned, was the motor that's thought to be climbing and slipping along the sides of the stereocilium. And so Peter Bar-Gillespie's group had developed a mutation within myosin 1C that made it susceptible to a specific ADP analog. And in the presence of this ADP analog, what it did is it basically froze the motor and didn't allow the motor to function, and froze it in an actin-bound state. And so what they found was that without the drug, you could see the slow adaptation process. But in the presence of the drug, you no longer saw as much adaptation, that the adaptation was largely diminished, suggesting then that indeed this myosin motor was participating in and important for the slow adaptation process. And so again, this led to the idea that the myosin 1C were the motors that were climbing and slipping along the sides of the stereocilium. And so one thing that they didn't do previously was the mechanical experiments in terms of using a force stimulation and measuring the hair bundle displacement. And so we wanted to do that. And so we do that now with a fluid jet in the mouse vestibular system. And we repeated those same experiments they did, but in this time we're using a force stimulation. And we found exactly what they had found previously, that the decrease, that we see robust adaptation without the drug, but in the presence of the drug, that slow adaptation is diminished, okay? But now additionally, we have this mechanical information, and this mechanical information told us that there was not much change again in the displacement of the hair bundle. Again, suggesting then that there's no correlation then between the slow adaptation and the hair bundle creep. So this was now another method of modulating adaptation, which didn't result in a change in the hair bundle motion. Okay, so this leads me then to the conclusion of the second part, where slow adaptation does not seem to actually involve myosin motors slipping along the sides of the stereocilium. And so based on this, we no longer think that calcium is actually coming in and affecting these myosin motors along the sides because the mechanics of the hair bundle don't change. And because of the fact that this creep is not changing, it also tells us that the tip link cannot be mediating the effect of slow adaptation. So this means then that the slow adaptation process likely has to do with calcium coming in through the channel and actually affecting the channel somewhere much closer to the channel, and is not actually affecting anything up here, because it's likely any effect up here would have to be translated through the tip link. And again, since the tip link contributes to the tip link tension contributes to the stiffness of the hair bundle, then we would expect to see that the mechanics would actually change, that the creep would actually change. So we think then that calcium affects the channels much closer to the channels in the slow adaptation process. And so now I'm going to end with just to summarize the 2 points again. So first, the first observation we made with short stimuli and a stiff probe is that we saw that that adaptation looked the same between negative and positive potentials. And this led us to conclude that fast adaptation is not actually driven by calcium. And the second observation is that with a fluid jet stimulus and where we can focus on slow adaptation, we did not see any change in the mechanics of the hair bundle, suggesting then that calcium was not actually mediating a regulation of the tension of the tip link, and therefore not mediating the climbing and slipping of the myosin motors. Okay, and then additionally, this means that that we think that the calcium is actually acting much closer to the channel for slow adaptation as well. And so what does this mean in terms of what we think is actually going on with these mechanisms? So we know that this fast adaptation is not calcium dependent. And so we think that it could be actually just a mechanical phenomenon. So one possibility is its viscoelasticity. And the idea of this comes from the fact that when we use the fluid jet stimulus, we actually mask the fast adaptation current response, suggesting it could be strictly a mechanical effect. And so we think that maybe there's some viscoelastic element within this mechanical system. Now for slow adaptation, previously it was thought that slow adaptation had the myosin motors at the tip of at the sides of the stereocilia had two functions. First, it acted in, it mediated slow adaptation. And the second was that it actually generated tension. And the reason that this resting tension is important in the tip link is this resting tension is important to allow us to hear very soft sounds, to give us this very low thresholds of sound. Because without the resting tension, then the channel is not very sensitive to very small amounts of motion. And so it's thought initially that these myosin motors acted both as tension generators as well as mediating slow adaptation. So in our new model, we think that there's a separation of duties. And so we think that there are still myosin motors that do tension the tip link. And we actually think these motors are myosin 7A based off a collaboration with Jungbom Shin's lab that we published earlier this year in Nature Communications. And so we think these motors are present and they generate tension in the tip link, but we do not think that they're responsible for slow adaptation. Now there's another molecule that's important for slow adaptation, and that's PIP2. So PIP2 has been shown to be important for slow adaptation and be able to modulate the properties of the channel. And additionally, more recently, Chris Cunningham and Ole Mueller's group has shown that a PIP2 interaction with TMIE can also modify the properties of the mechanotransduction channel. So we think that it's possible that PIP2 may hold the key towards the adaptation mechanism. And so we think that there's a second set of motors that are responsible or important, I should say, for slow adaptation. And these myosin motors may be important just for either concentrating or transporting PIP2 towards close to the channel. And this is based on studies that have shown that myosin 1C can actually bind to PIP2. Right. So with that, I'd like to finish by thanking my lab members. So Juicy is a postdoc in the lab, and she has done most of the work on the slow adaptation work that I presented today. Andrew Mecca is a graduate student in the lab, and he also contributed to this work, as well as other work that I wasn't able to talk about today. Additionally, I'd like to thank our collaborators, Jungbom Shin's lab, as well as Tony Ritchie, and also Peter Bar Gillespie, who gave us the myosin 1C mice, as well as our funding. And to end things, I want to just put a little plug in that there is a postdoc opening in the lab. And so if anybody is interested in this type of work, I would be excited to hear from you, since we are looking for more people to be joining the lab. So thank you all for listening, and I can start to take your questions.
Thank you very much. This is a great talk, Anthony. And so while the questions are coming in, I have a couple of questions. If you could maybe elaborate a little bit. So you did a very good job of summarizing different species and all the previously published data that it came, some came from, some results came from frogs, some from turtles, and all of your results are from rats and mice. So the question I have is, so are you planning on trying to, so what previously happened is obviously the results from lower vertebrates were used to explain more about what might be happening in the mice, because technically it was much more challenging to do these experiments in mice in the first place. So now that you have the results in mice, how do you view this entire pool of data? Do you think the mouse results that you get are, even somewhat related to what might have been related to frogs and turtles, or are they just completely different in terms of their adaptation mechanisms?
So that's a good question. I think for fast adaptation, I think it's still a little bit unclear, because I think the data in terms of fast adaptation seem to be pretty convincing in the bullfrog as well as the turtle, suggesting that maybe fast adaptation may have a different effect of calcium. Now, it's not to say that calcium doesn't affect fast adaptation. Calcium seems to modulate the fast adaptation process and can modulate it slightly, but it doesn't seem to drive it, at least in the mammal. And that may be the case as well in the turtle, and because there's some indication that at positive potentials, it may open probability of the channel, so kind of the resting current of the channel, changes greatly in the turtle, whereas it doesn't so much in the mammal. So it's possible that that mechanism does actually translate over to the turtle as well, but it's basically masked by some other properties of the turtle that is not present in the mammal. Now for the slow adaptation stuff though, I do think that this is a result that actually spans across species. And this is because actually Crawford and Fetty Place published, I think it was either in 1989 or 1991, that they similarly did not see a change in the mechanics of the hair bundle when they depolarized the cell. And that was the only other instance where this type of experiment was actually performed. Because the original work by Howard and Hudspeth didn't actually do this type of manipulation. So I do think that that result does translate across species. And additionally, since the vestibular hair bundles also seem to exhibit the same type of thing, which is also much closer, let's say, to what the kind of bullfrog and turtle hair cells are.
Thank you. So there's one question that is somewhat related. Great talk. Given what you revealed today, what do you think is the mechanism for shifting the operating point? Does it depend on calcium? And does it depend on myosins?
So yeah, so I do think that, so there is a calcium mechanism that can shift the operating point, because we know that low extracellular calcium, and so this was work that we published in the journal Neuroscience in 2016. And so we know that extracellular calcium can regulate the operating point of the channel. But we think that this actually happens separate from slow adaptation. So I think there are multiple mechanisms that can actually regulate the operating point of the channel. And one of these, and we proposed that this was potentially, for instance, through the lipid membrane, based on some work with using GSMTX4. So I think that calcium does regulate the operating point, but it may happen through other mechanisms. And obviously slow adaptation also is calcium dependent and also is regulating it, but just on a different time scale than maybe what some of these other things are working at.
Okay, another question. Amazing work. So how does this impact our concept of spontaneous bundle movements in non-mammals and possibly in mammals?
So that's a good question. So obviously spontaneous bundle movements are still present, and that doesn't go away. And from my understanding is that spontaneous bundle motion really just needs some power generator, okay? And so I think that power generation may be just due to this myosins climbing up along the sides of the stereocilium, right? And then this release of tension, which may regulate the channel due to, this change in tension is not actually happening, let's say, in the process of slow adaptation. Okay, so that there's some power generator, and those power generator comes from the myosin motors, but then the adaptation mechanisms maybe work to create the bundle oscillations through a different mechanism. And so I can't explain all of that yet, and since I haven't done experiments to look specifically at those processes.
Thank you. Another question. If you somehow affect slow adaptation in a mouse, say genetically, how do you think it will manifest in its hearing performance?
So that's a really good question. I think it's basically getting at what is the physiological function of slow adaptation. You know, in the mammalian system, slow adaptation is potentially happening outside the frequency range to, let's say, contribute to the kind of bandpass filtering that I mentioned before. So slow adaptation may be playing a role on a slower time scale than let's say, than tuning, and act more to adjust the operating range, let's say, and adjust the range of sensitivity. And so what would it do to a mammal? And I think that's a great question, and it's something that I would like to answer and get an answer to, but I can't actually give you that answer yet. But my guess is hearing performance would be degraded, but I can't give you specifics in terms of exactly how. That is, I think that is a key question that that we still need to answer. You know, what is the physiological relevance of these adaptation processes?
Oh, here's one technical question. Super talk, in your high-speed videos at the highest stimulation frequencies, how do you deal with the heating of the preparation with the high light levels?
Oh, so it's a good question. So again, so I mentioned the high-speed video, but it's actually a combination of things that have allowed us to do what we do now. So number one is high-speed video, but the second is actually high-intensity LED lights. So high-intensity LED lights actually don't have the issues that our regular kind of microscopy lights have, which is heat generation. So general, let's say, kind of the incandescent light or even the metal halide, those types of lights also generate a lot of infrared wavelengths. And those infrared wavelengths is what actually leads to the heating of the tissue. And so with, again, the increase in power of LED lights that are much more narrowband and don't give off that amount of heat, you actually don't get tissue heating. And so we've actually measured the amount of the temperature change, let's say, in the dish with our high intensity LED lights, and you maybe get 1 degree Celsius or at most 2 degrees Celsius change. So this high intensity LED lights is actually a second piece of the puzzle to be able to do this high speed imaging.
Right. Okay. So another question. We have a few more minutes, so I don't think we'll be able to address all of them. But so I'll rephrase this question a little bit, but it is related to myosin 1C results that you presented. So do you want to elaborate a little more about the myosin 1C result and say, you know, there was a little bit of residual adaptation in your slide on the vestibular system. Do you think myosin 1C is still playing some role in vestibular and potentially the cochlear hair cells in mice?
Good. So good question. So in the vestibular hair bundles, we do think that myosin 1C is playing a role. So another experiment that I didn't talk about today is that we also repeated these experiments in the cochlea. And so it doesn't seem that in the cochlea, myosin 1C plays the same role that it does in the vestibular system. It could be that there's a different myosin motor that's involved in this process in the cochlea, but we do know myosin motors in general, since with our inhibition experiments, we do see that adaptation is reduced. So myosin motor activity is indeed important, but it just may be a different myosin species, let's say, that's important for that. So there does seem to be differences, and there are differences, let's say, between even the molecular players between the vestibular system and the auditory system, and there's obvious mechanotransduction characteristics that are different between the vestibular and the auditory system as well. So there are definitely different mechanisms potentially at play.
All right. One question related to the tip link. Thanks so much for your amazing work. Recent structures and simulations of stage 15 shows both elastic and extensibility as the gating spring. Are those properties the missing part of slow adaptation? Can you elaborate whether the elasticity basically of the tip link plays any role in slow adaptation?
So I don't think that actually elasticity in the tip link is playing a role in adaptation, and that's because of the fact that the mechanics of the hair bundle don't change. As I had mentioned, that the tip link contributes to the stiffness of the hair bundle, and specifically, it's really the tension of the tip link that contributes to the stiffness of the hair bundle. Such that if you change the mechanical properties, and let's say you decrease the tension by allowing this elastic element, I would expect then to see a change in the motion of the hair bundle during the stimulation. And since there's no correlation between the displacement of the hair bundle and the amount of slow adaptation, I actually don't think this kind of tip link elasticity plays a role there. Now, where it could play a role though is in fast adaptation. It's definitely possible that fast adaptation, for instance, could be involved or could be related to some of these kind of elastic elements that may be in the tip link.
Well, as a follow-up on that, do you think then your imaging is just unable to resolve? Because you would expect that even if it's involved in fast adaptation, you would still see some kind of related bundle motion, right? So is imaging too slow to visualize that?
So no, because if you look at the fast adaptation, so we do see that bundle relax or that mechanical creep. And so essentially that mechanical creep could be some viscoelastic element. So potentially, for instance, that element could be in the tip link, right? And so if this mechanical, because in that case, it's a mechanical motion, and we're not modulating or modifying the amount of fast adaptation at negative and positive potentials, which is why that mechanical motion may be the same in both cases.
Okay, it just has to be calcium independent then.
For slow adaptation, it would have to be calcium dependent, but not fast adaptation.
Okay, thank you. So
maybe we should go to the last question, and then again I can continue to answer questions via email as well.
Yeah, I can't see any questions you can quickly answer. Let me see which one is. How do you think this contributes to hearing in a mammal receiving high frequency sounds, say 40 kilohertz?
Sorry, repeat that.
How do you think this contributes to hearing in a mammal receiving high frequency sounds, say 40 kilohertz?
So 40 kilohertz. So I think, again, I think this gets at what the physiological relevance is. Now, we propose that, that the physiological relevance of adaptation in general could increase the dynamic range. It controls, the sounds that we're sensitive to, as well as participating in frequency tuning. Now, for the slow adaptation process, again, like if slow adaptation is too slow for those high frequency sounds. And so that, in that case, I think it's probably contributing more towards dynamic range and the operating point, and setting basically what levels of stimuli that the hair cell may be sensitive to. But I don't think it's playing a role in the frequency tuning likely. So I think, again, though, these types of experiments, it would be, one of our goals is to basically specifically, let's say, knock out or inhibit the slow adaptation process to really see what happens to, let's say, ABRs or frequency tuning and those types of things in the cochlea. So.
Okay, well, thank you everyone for attending this seminar, and thank you, Dr. Peng, for giving an amazing talk. And I also want to thank the ARO for organizing this event.
Yes,
thank you, everyone.
And thank you, everyone, for coming again. And then also remember that Jennifer Stone will be talking in a few weeks as well. So be sure to tune in for that. Thank you.